Hi everyone, my name's Lycia and I'm a part of team of Mice and Ming. My team members are Sue, Artie, and Tien. During our time at the Neuromatch Academy, we looked at predicting mice behavior and assessing information propagation using spiking patterns. When we carry out behaviors, neurons in the brain exhibit very variable activity, but we still don't know how populations of these neurons connect and communicate with each other. The aim of this project was to identify which neuronal features might be good predictors of successful and unsuccessful behaviors in mice. We chose to use the Steinmetz dataset to address our research questions. It contained recordings from different brain regions in mice carrying out a contrast discrimination task.
In the next part, I will be talking about how we process the data. For our first aim of using spike data to predict the mice behaviors, we have conducted several steps to filter and reduce the dimension of the data. Firstly, we filter out trials in which there were no contrast difference and hence no contrast discrimination. Secondly, we group neurons into inhibitory and excitatory neuronal types based on trough to peak data. And thirdly, we also filter out non-responsive neuron by counting the spikes that happen at or in close temporal proximity to the three important events within a trials, which are cue onset, response onset, and feedback onset. And then lastly, we also group neurons to their respective brain areas. And in this data set, we have 8 brain areas. We're showing some graph of spiking rates that has been averaged across all trials and across all neurons of the same brain area in selected sessions for one mouse.
After the rigorous filtering process, we were able to reduce the dimension of over 1,000 neurons to 16 columns, each column representing one of 8 brain groups split into excitatory versus inhibitory neurons. We trained the logistic regression model with these filtered brain group data with only event-relevant neurons as input and the mouse performance as output using L2 regularization. Because each session had different brain areas as shown in the brain area section above, logistic regression had different coefficients for different sessions. With that caveat of each model only representing brain areas that particular session had, each model had a consistently good predictive accuracy for the mouse task performance. In terms of hyperparameter C, we used nested cross-validation since we saw better performance with nested cross-validation as opposed to non-nested cross-validation in using our own Steinmetz dataset. Here in the next section, we are showing the basis as to why it's important to distinguish excitatory and inhibitory neurons in the input data. As you can see in this sample graph of session 0, the excitatory neurons of the visual cortex in the top graph have spikes that precedes inhibitory neurons in the bottom graph.
Here we present the leaderboard of performances of each mouse for the sessions they participated in. As you can see, there's a lot of variability in their performances. Best performing mouse was Liederberg and worst one was Moniz. For each of these two, we prepared predictive model and plotted the model accuracies for their sessions. Liederberg model accuracy was 80 to 90% and Moniz model accuracy was 75 to 80%. In conclusion, spiking data seemed to be a good predictor of behavior. We studied information propagation and observed excitatory neurons preceded inhibitory ones. We also observed a lot of variability in each mouse. Future work includes expanding latency window for all brain areas to study functional connectivity and compare connectivity between best and poor performing mouse. Also analyze waveform data and local field potential data in the original data set. Thank you for your attention.